I'm absolutely delighted to be here. I was thinking on my drive to San Diego from Los Angeles, when was the last time I was here to give a scientific talk? I do visit San Diego. It's a beautiful place to live. You're very lucky to live in such a gorgeous location. But I actually haven't been here very recently to talk about our stem cell work. So I'm thrilled to be back. If it's been 20 years, that's terrifying. It went very, very fast. But I'm thrilled to be here. So I'm a basic scientist, and I build knowledge on how cell lineages form in the embryo and use that knowledge in order to engineer cell types and organs from stem cells, and in particular using pluripotent stem cells. So today what I plan to do is tell you about the organ that we've been spending a lot of time thinking about and the cell lineages in that organ that we are generating from pluripotent stem cells, and how, in order to achieve this, we need to work between a number of different model systems in order to understand the development, the ontogeny of these lineages in vivo during development, and then apply that knowledge to the differentiation of those cell types in the lab. So what we do with the organ that I'm going to tell you about today is very similar to what most stem cell scientists in their labs will do, whether you're trying to create a brain organoid or a kidney organoid. It's that interplay between understanding the developmental biology of the organ and then applying those principles to in vitro differentiation. So the organ that I'm going to tell you a lot about today, and the cell types that I'm going to be talking about, is the ovary. So the ovary is an organ that's found in females, babies assigned female at birth. I'm going to use the term women and girls when I talk about the ovary, but I acknowledge that not everybody represents their gender that way, and so I want to acknowledge and represent that, but I'll be talking about women and girls and the ovary. So this image of the ovary, we can see in blue are the germline cells. And so at the cortex, so this is the outer cortex of the ovary, we have germline cells that are arrested in prophase 1, end of prophase 1 of meiosis 1. These are marked by blue through a protein called VASA, which is specifically expressed within the adult germline. These oocytes are surrounded by a somatic cell called granulosa cells. I'm going to talk about granulosa cells today as well. These granulosa cells that surround the oocyte make up a unit called the follicle. And these follicles that are found underneath the cortex of the ovary are called primordial follicles. So when you hear about the term ovarian reserve, what the ovarian reserve refers to is the number of these primordial follicles that are found under the cortex of the ovary. So each cycle, and in women it is about once every 28 days, a cohort of primordial follicles are recruited into follicular genesis. And what that means is those granulosa cells, which are surrounding these oocytes, will start to change shape, they will start to proliferate, and they will start to change in character and give rise to activated follicles. So most of this organ that you can see here is composed of activated follicles. And these structures are made up primarily of proliferating granulosa cells. Every one of these follicles does contain an oocyte, although you can't see it. This is a section, a histological section through the ovary. These follicles are very large, and so in some of these sections, for example here, the oocyte is just not in the section, but it would be beneath it or above it, depending upon how it's being cut. So these follicles are what's called the functional units of the ovary. So as a scientist trying to model ovarian biology and ovarian development, we need to think about the development and differentiation of the germline, which is the generation of the oocyte, as well as the development and differentiation of these granulosa cells, not only the ones that surround that primordial follicle, but how they change through development to form the activated follicles. This is important because the ovary has two functions. One, it is to provide reproductive capacity to that person. The germline is the only cell type in the body that's able to pass DNA from one generation to the next. Once all these germline cells are lost, this person no longer is able to have a biological child that's genetically related to them. These granulosa cells in the ovary are really important because they provide the endocrine function of the ovary. I think perhaps that's a little discussed function of the ovary, but it is really important to supply estrogen, progesterone, and testosterone, which are essential for overall health and wellness. And so when these follicles disappear, that is when estrogen is no longer produced, progesterone is no longer produced, and testosterone is no longer produced by the ovary. And we all know that the loss of the follicles occurs with a phenomenon that's referred to as menopause in women. So what drives menopause actually is the number of follicles that are established during fetal life. So these primordial follicles out here, they are established prior to birth. From what we can tell as developmental biologists, there are no germline stem cells in the mammalian ovary. And this is very different from other organisms, including other vertebrates such as zebrafish, where the germline does develop continually from a germline stem cell population. So in mammals, there is no germline stem cell population as far as we can tell. Instead, the germline is composed of these post-mitotic meiotically arrested oocytes. So as these are recruited, that pool of oocytes, pool of primordial follicles, the ovarian reserve, is progressively diminished. So if scientists can figure out ways to prevent the activation of these follicles into the process of follicular genesis, that would be a way of delaying the loss of the endocrine support of the ovary. So as these follicles are recruited out of the ovarian reserve and into follicular genesis, and as these follicles start to grow and communicate with the oocyte and begin to produce hormones, such as estrogen, most of these follicles will die. So this is another concept that occurs in the mammalian ovary, is that there's a large recruitment of follicles and most of them will disappear. And in women, only one of those follicles becomes the dominant follicle and an oocyte is released. So there's a lot of attrition of follicles that occurs in the adult ovary, and that's also what drives the loss of ovarian function, the loss of endocrine function, the loss of reproductive capacity. So I wanted to line up follicular genesis in a different way so that you could see it. This is an image that comes from a review that was written by Melina Schul's group who studies the oocyte. And so basically, to summarize what I just said, the ovary is composed of these primordial follicles, an oocyte surrounded by around 4 to 6 granulosa cells, and that these follicles are gradually lost from the ovary through activation into follicular genesis, which creates primary follicles, which means one layer of granulosa cells around the oocyte, secondary follicles, which means 2 layers of granulosa cells around the oocyte, so proliferation of the granulosa cells, and then ultimately multi-layer follicles, antral follicles, and then the release of the oocyte from the ovary. And these granulosa cells during this window are producing primarily estrogen, and after the release of the oocyte, those granulosa cells will go on to produce progesterone. So the other important aspect of the ovary is that it ages faster than any other organ in the body. So in my lab, we don't study ovarian aging, but the developmental principles that we're learning about the ovary can actually inform about the process of ovarian aging. And so this is essentially a graph showing how organs in the body age, and you can see that the ovary essentially becomes non-functional in women at around 50, loss of the germline, no longer produces hormones because of loss of those follicles, compared to other organs in the body which age much more slowly. And so a new field has emerged in ovarian biology starting to think about this concept of ovarian aging and how to slow the process of ovarian aging down so that the age of the ovary can be more consistent with the age of a human being. The aging of the ovary is important and consequential. So when the ovary ages, this leads to different susceptibilities to disease. So for example, after the ovaries stop functioning, and whether it be over the age of 50, which is normal menopause, or for those individuals who suffer from early menopause, this leads to to challenges with the cardiovascular system, changes in the immune response, more brittle bones, so osteoporosis is increased in women whose ovaries have stopped functioning, as well as cognitive decline due to lower levels of estrogen. So thinking about how the ovary forms, thinking about the ovary as an endocrine organ, not just as a reproductive organ, is really important for overall health and human health. The other important aspect to think about the ovary and ovarian function is how society is changing in terms of family structure, as well as when individuals are choosing to start their families. And this is a graph from the Centers for Disease Control looking at birth rates. So this is from the point of view of women, but looking at birth rates of first child in the 1990s compared to 2019. So in the 1990s, most women were starting their families in their 20s. So you can see here that this is the largest group of when women are having their first child. 30 years later, we can appreciate that this is changing tremendously. In fact, the fastest group that are having their first child, the fastest growing, or the largest change, I should say, is in women who are in their 40s to 44 age group. So now at this point, we are hitting up against the loss of that ovarian reserve, the loss of follicles, the loss of hormonal support, the capacity to be able to carry a baby to term successfully, but also the loss of the germline, which is essential to have a biological child. And we can see that the two other fast-growing groups that are changing from 1990 to 2019 are women in the 30 to 40-year age group. And I certainly am one of those. I had my first baby at 35. I had my second baby at 40. And so this is very consistent, particularly with women who are delaying childbearing so that they can have careers as well as have a family. What this means is that there is increasing use and access to the assisted reproductive technologies, because having a baby at 35, at 40, at 45 can be a challenge. And so therefore medical support is required for some women to be able to do this. The other aspect of thinking about fertility, of ovarian biology, of reproductive biology, is that there is a significant inequity in individuals who suffer from these diseases, but also in their ability to access reproductive care. And so this is something that my lab also thinks a lot about now as we're building models of the ovary, and we're thinking about how these models can be used to understand development and disease. Is to build models that are more representative of a diverse population and not just studying ovarian biology, oocyte biology, reproductive biology in one race and one ethnicity. So in the United States, Black women are nearly twice as likely to experience infertility than their non-white Hispanic counterparts. And Asian women in the United States are least likely to access assisted reproductive technologies early, meaning as they're delaying their childbearing, this is again leading to problems with their fertility because of the ovarian clock. So how can we reduce the burden of ovarian disease and reduce the burden of infertility? So the most important thing is to generate new knowledge on reproductive organs. And in our case, we build new knowledge on mammalian ovarian biology, understanding the cell and molecular regulation of how those primordial follicles are built and how those follicles are activated in the process of follicular genesis. Most of the work in ovarian biology is performed in the mouse as a model, and with very little ability to study human cells. And that's because access to human reproductive tissues, in particular access to human ovaries containing follicles, is very rare. The only way to access these tissues is if women are seeking assisted reproduction and preserving ovarian tissue to be used later after cancer therapy or chemotherapy agents. And those tissues are very difficult to come by research. So generating human ovarian models using human cells would be a very important model for the reproductive science community. As we're thinking about toxicology, there's a lot of evidence to suggest that the oocyte and the ovary are exquisitely sensitive to environmental agents, that we need models to be able to screen environmental agents for their effects on the ovary and effects on the germline. And so we believe that pluripotent stem cells can create these in vitro platforms that are amenable to screening. Understanding environmental impacts on the ovary and the germline means that we need to be able to study the ovary and the germline in utero. So how is the ovary created during embryonic life, during gestation? How does the ovary change during puberty? Because this is when you move from the activation of those follicles and the degeneration in the center of the ovary to the ability of those follicles to become ovulated. And then also in the reproductive years, so access to tissues with follicles. When I first started working in the stem cell field, this was at UCSF with Renee Reopera. So when I was at Baylor College of Medicine, I went there because I wanted to learn mouse genetics. I wanted to make genetic models to understand reproductive development and disease. And that was a fantastic experience. But what I realized as a postdoc back in the early 2000s is that if I was interested in translating any of this knowledge from mouse to human, then I needed to start thinking about human models of development. So I went to Renee Reopera's lab, who was working with embryonic stem cells at the time that were from Wisconsin. So these are Jamie Thompson's lines, as well as some of the lines generated by Roger Peterson at UCSF. And I put this picture in here because this is a picture I took as a postdoc down the microscope, and you can see phase contrast microscopy has not changed much in 22 years. I took this picture, at least what it said on the slide in my computer was I took it in 2004, so it's 20 years old. This is the H9 line of human embryonic stem cells, and this is growing. So this is a colony of human embryonic stem cells, so we all know that by now. And at that time in the early 2000s, we were growing pluripotent stem cells. There was no such thing as iPS cells. They hadn't even been thought of yet. Perhaps Shinya had thought of them, but at least they weren't they weren't available or human. That these pluripotent stem cells were cultured on a mouse fibroblast feeder layer, and that's how the field cultured human embryonic stem cells, at least over the next decade. And the initial human-induced pluripotent stem cell lines were also derived and cultured on these feeder layers. And so what this meant was that there was a lot of variability in our differentiation experiment. So as we were differentiating cell types in vitro, the same line would give us cell types, but the fractions of those cell types would change slightly. And so we never knew whether these differences between cell lines were due to the fact that we didn't know how to grow human pluripotent stem cells appropriately. Were these MEF layers contributing differences in the self-renewal and pluripotent capacity of these cells? There was a lot of unknowns about the MEF-dependent system. So now we culture our human pluripotent stem cells feeder-free, as does, for those of you in the room working with pluripotent stem cells, I'm sure that all of you do as well. And there's a lot of different feeder-free ways to culture human pluripotent stem cells, and we use a variety of them, and all of them are excellent. This is an image that my undergraduate took of one of the UCLA human embryonic stem cell lines that we grow in the lab. This is in StemFit laminin. We like using the StemFit media because it's very stable with single-cell passaging. So it enables you to culture human pluripotent stem cells like you would with mouse pluripotent stem cells, and they remain karyotypically stable. Because it's a single cell way of passaging, it enables you to genetically modify them and create reporters very, very easily. So this is a media that we've been using a lot. So if you want to make organs in the lab, you make organoids. And so organoids are made by a variety of investigators. Many of you are in this room generate organoids in the lab that are either coming from embryonic stem cells or induced pluripotent stem cells that originate from blastocysts or by reprogramming. And these organoids are organs that are generated from specific germ layers. So we have the endodermal organoids, which include the gastrointestinal, liver organoids, lung organoids, etc. We have the mesodermal-derived organoids, which include the kidney organoids. And then if we have the ectodermal-derived organoids, which would include the entire suite of brain organoids that exist. In the ovary, we've got to think about this differently, because the ovary is not made from ectoderm or mesoderm or endoderm. The ovary is actually made from mesodermal derivatives and the germline. And the germline is from a completely different germ layer. So the way we have to think about building these organs is to think about building tissue layers separately and then combining them together. And so the terminology that's used to describe this was first coined by Sergi Pasca, and I really like this terminology. I think it fits very nicely for the work that we do in the ovary, and that's the assembloid technology, which is the idea that you take cells differentiated one way or tissues that are acquired one way, and you combine that together with tissues differentiated a different way or acquired a different way, and then you combine them together so that they can self-assemble into a three-dimensional structure. That's essentially what an assembloid is. And so therefore, when we're thinking about building ovaries in the lab, what we're building is not organoids, but assembloids. And so this is an assembloid of the ovary. This is an ovary that's made entirely in vitro. And so we call it an assembloid because the germline is derived from induced pluripotent stem cells. And these induced pluripotent stem cells carry a reporter tool in them, which is CFP, so a blue fluorescent protein, which is driven by a germ cell-specific reporter called STELLA, also referred to as DPPA3. And so you can see that this is an oocyte. This oocyte is surrounded by granulosa cells, which are not stained here, so they're seen in grey. And these granulosa cells, in this particular example of an ovarian assembloid, are originating from fetal tissue. So the somatic cells are coming from fetal tissue as a single cell suspension. The germline is differentiated in vitro from stem cells. We combine them together in low adhesion plates. They self-assemble, and then over the next 3 weeks, they will form this ovarian model, which has critical cell types we can use to understand ovarian development and disease. Now, this model of the ovary is a mouse model, and so we make these models all the time in the lab. So I have technicians in the lab whose sole purpose it is to make model after model after model after model, and we use them to study development and disease, but also as the framework to be able to create this same model using human cells. So I want to take you now through how we're thinking about making the ovarian model with human cells. So I told you the ovary is composed of the germline and the ovarian somatic cells. So I need to tell you in development when they each form. So this is showing development from the point of conception or fertilization all the way just prior to birth, which in humans, as we all know, is 40 weeks. The germline is actually one of the first lineages to form in the embryo. It forms between week 2 and week 3 post-fertilization, and that's called the specification of primordial germ cells. And as Evan said, I've spent a lot of my career studying the specification of primordial germ cells, because if you don't specify them, you're not going to be able to make a functional ovary. So they're very, very critical to the function of the ovary. Then about 2 to 3 weeks later, we have the induction of ovarian somatic cells from the intermediate mesoderm. And these ovarian somatic cells are first seen at around 5 to 6 weeks post-fertilization. So we're looking at a number of weeks different between the specification of the germline and then the specification of ovarian somatic cells. Then at around 20 weeks, we begin to have the initiation of follicle formation. The follicles are the meiotic oocytes surrounded by those 4 to 6 granulosa cells that form the ovarian reserve. They begin to form at around 20 to 25 weeks, and then by birth, all of those primordial follicles have formed. So there's not thought to be any more genesis of follicles. Follicle formation is complete and the ovarian reserve is fixed. So you can imagine that if you specify fewer primordial germ cells, if there are problems with the specification of the ovarian cells, if there is issues with the establishment of follicles, this all impacts the number of follicles in the ovary, and that'll dictate the aging of the ovary. Lose those follicles, ovary is now non-functional. So the questions that we've been asking in the lab is, where does the human germline originate? This is a simple question for which there is no simple answers. And so the human germline forms around 2 to 3 weeks post-fertilization. And that is at a time where it's very difficult to access human tissue, because it's before a woman would even realize she's pregnant, even if she wants to terminate the pregnancy, and the tissue gets donated to research. So it's in what scientists refer to as the black box of embryo development. And so what we decided to do was take advantage and leverage the technologies that were being generated by Magdalena Zinitska-Gertz's lab and Ali Bravanelou's lab, which is to culture human embryos up to the limit of culture, which in the state of California is actually 12 days post-fertilization, which is different to the international consensus, which is a social contract we as scientists have with the public that we will not culture embryos longer than 14 days or emergence of the primitive streak, whichever comes first. Why we have 12 days in California is a little bit of a mystery to me, but it was written into the proposition, and that's why it's in California state law. So the first proposition as well as the second proposition both have the 12-day rule. So we began a project where we consented human embryos to research for embryonic stem cell research and also to understand early development to differentiate these cells in vitro. This is a human blastocyst at day 6 post-fertilization. Most of the cells you can see here are trophoblast cells in blue. In green, what you can see is the pre-implantation epiblast cells. These are the ones that are going to go on to form the embryo or embryonic stem cell lines. And in red are the SOX17 positive primitive endoderm cells that are going to form part of the extraembryonic membranes and potentially even some of endoderm inside of the embryo. But mostly these SOX17 positive cells form parts of the extraembryonic membranes. So you can appreciate the inner cell mass is not just pluripotent cells. The inner cell mass is made up of 2 cell types. It's the pre-implantation epiblast as well as the SOX17 endodermal cells. So in order to study the post-implantation window of development, what we do is take these blastocysts and put them into what we call attachment culture. And these blastocysts self-assemble in order for the endoderm to move around. So instead of being in this salt and pepper pattern, the endoderm will move around the epiblast, and the epiblast will form the amniotic cavity. And so to show you what that looks like, I want to show you here. So this is an attached embryo at 12 days post-fertilization. So you can see the trophoblast, which in this case is shown in pink, has expanded and flattened down on the plate. That epiblast, which in the previous slide was green, here it's blue, has also expanded. So there's more than 10 to 15 cells, and the epiblast has formed a cavity, which is the proamniotic cavity. In addition, the SOX17 positive cells have started to surround this epiblast to form what would be the primitive endoderm. So this is the self-organization process of the attached human embryo cultures. And these were used to essentially demonstrate that the amniotic cavity, when this can be pushed to 14 days, the yolk sac cavity does not require a uterus in order to do this. So this is a self-assembly process that can occur entirely by the embryo in vitro. So we wanted to know, do human germ cells emerge within these first 12 days of culture? Can we use the human embryo attachment culture model to study the process of early human germ cell development? And so to do that, we thawed out more than 100 human blastocysts. So this process of attachment culture is very, very inefficient. And so, but we were able to find 2 out of the 25 embryos that maintained the epiblast, the endoderm, and the trophoblast. We were able to see evidence of germ cell formation. So how do we know they're germ cells? We know that germ cells co-express the transcription factors TFAP2C, which also mark the trophoblast. They express nanog, which alone marks the epiblast, and they express SOX17, which marks the primitive endoderm. So this staining strategy enabled us to identify early germ cells as being triple positive for SOX17, TFAP2C, and nanog. So you can see rare cells, one rare cell here that I'm pointing out. So in humans, what this tells us is germ cell formation begins soon after embryo development. It's one of the first lineages that emerges after the embryo implants. But because we can't study human embryo development longer than 12 days in the state of California, we can't study human germ cell development effectively using this model. So we need alternate models to be able to do that. So the alternate models that we use in the lab currently are shown here. So we use something called three-dimensional disorganized aggregates, which that means taking human pluripotent stem cells, putting them in low adhesion plates, adding cytokines we know are essential for the specification of the germline, in particular bone morphogenetic protein 4, and letting differentiation progress in this disorganized way. It's in 3 dimensions, but it's disorganized. We can generate a lot of primordial germ cells using this model. So we use it when we need a lot of germ cells. What we have been doing over the last few years is working in the embryo model space in order to study the three-dimensional patterning in the early human embryo and the specification of the germ cells. So one of the models that we worked on with Jianping Fu is the embryo model called the PACE model. This is a three-dimensional model that specifically models amniotic sac formation, and what we find very nicely is the specification of the germline. So when we're looking at these models, the germline is specified and can first be identified within the amnion layer, so in the extraembryonic membranes. And we can also see, although not shown in this image, germ cells in the pre-implantation epiblast as well. So our hypothesis is that the germline can originate from two sites in the early embryo. And we've started to work on two-dimensional models of germline formation so that we can apply this to live imaging to study the emergence of the germline. Not much is known about early human germline development. And so as we were building these models from stem cells, we wanted to be able to profile and understand the molecular integrity of the germline as it's forming. And so one of the ways that we did this was to take that disorganized aggregate model, which is a two-step differentiation, starting with human ES cells. We then exposed them to activin A and Chiron to make this cell type called an incipient mesoderm-like cell, where some of the cells have progressed into mesoderm, but other cells have actually started to head backwards in development to more of a naive-like state. We think that they're the ones that give rise to the germline. And then we expose those cells in three-dimensional aggregates to a cocktail of cytokines. Using single-cell RNA-seq, what we're able to see is the germline in this model emerges around 24 to 48 hours after the BMP4 exposure. So we can start to see the beginning of germline cells here, and then there's a tenfold increase in the germline. This is additional specification of the germline that occurs over those next 24 hours. So these germ cells that we make in vitro up to about day 5 of those three-dimensional aggregates are equivalent to what we now appreciate as being Carnegie stage 7 of human embryo development. And we were able to achieve this by taking advantage of a reference map that was created in Cambridge in order where single cells were profiled of the Carnegie stage 7 embryo. This is around day 17 post-fertilization. And so when we take germ cells made from these 5 different iPS cell lines in the lab, and where we did single-cell RNA-seq and compare it to germ cells from that embryo versus germ cells that are found later in development, what we find is that our germ cells cluster closer to the Carnegie stage 7 embryos, early newly specified germ cells, compared to the later germ cells that are found at week 13. So when we line up the germline trajectory of these very early germ cells, starting from human ES cells all the way through when this experiment finished, which was day 4 of aggregate differentiation. So this is only cells that are in the germline and not any of the other somatic cells that are forming at the same time. What we identified was that in the early progenitors to the germline at day 1, so these are cells that have been exposed to BMP4, but they haven't fully committed to germ cells yet, which occurs at that day 1, day 2 transition, we find that there is an increase in the expression of what we call lineage primed genes. So these are genes that are not expressed in the specified germ cell, but they are expressed in that precursor to the specified germ cell. And these include transcription factors that many of us are familiar with, such as EOMs and brachyury, which are classic primitive streak markers, CDX2 and GATA3, which actually are classic trophoblast markers, as well as TFAP2A, which is a marker of the amnion. And so as the germline is forming, you're seeing an increase in these markers of different lineages, which is why we didn't define it as a cell type, but rather as lineage markers. And then they are rapidly repressed at the point of specification. When we line up the different transcription factors that are expressed over this time window, what we were able to come up with is signature transcription factors for each one of these time points in the germline trajectory from human ES cells all the way through to specified germ cells. So they're signature transcription factors and they're predicted regulators. And when we were doing these studies, we were very interested in the transcription factor TFAP2C because, one, we had used it as a marker of early primordial germ cells, and two, that it was starting to emerge in this day 1, day 2 transition as a key signature transcription factor, together with SOX17, which was shown to be an important regulator of germ cell formation by Azim Sarani's group, as well as Jakob Hanna's group together. And TFAP2C seemed to sit at the center of this transcriptional network. So in order to determine the function of TFAP2C in early germ cell specification, we use CRISPR and deleted TFAP2C in the prime state of pluripotency, where it has no function. And then we put the pluripotent stem cells through a differentiation protocol. And what we're able to show is that TFAP2C transcription factor is required for germ cell specification. So this is our percentage of human germ cells that we identify using surface markers that are specific to germ cells. We find that at day 2, there is around 10% of the aggregate is made up of specified germ cells, and this is remains constant at day 4. And you can see that in 2 independently created CRISPR mutants for TFAP2C, we make no germ cells whatsoever. And so therefore we're able to use these in vitro models to be able to ask questions about germ cell formation and the transcription factors and transcription factor network required for germ cell development. And that's what a lot of students in my lab do. So the germline is one part of the ovary. So now I want to talk a bit about human ovarian somatic cells and where do they originate from. I wanted to put this image up first because it's a really beautiful representation of sexual dimorphism in embryo development. So what you're looking at here on the left is the developing ovary in humans at 7 weeks post-fertilization. It looks nothing like the adult ovary. And here is the testis at around 7 weeks post-fertilization. The ovary and the testis originate from the exact same cell type. It's the genital ridge, the intermediate mesoderm becomes the genital ridge, and then you have sex determination, which will give rise to either an ovary or a testis. And so what drives this decision to form an ovary or a testis is based upon the expression of a very small number of transcription factors, and mutations in those transcription factors are what leads to disorders of sex development, or DSD. So here, the ovary, we can see that these here in pink is the stromal cells of the ovary, which is where the precursor cells for generating hormones are going to come from. And then over here are these cyst-like structures. You can't see them, but they're full of germ cells and granulosa cells that are intermingled with each other, and they have not formed follicles yet. In comparison, the testis, very different looking organ. This is the stroma, it's now shown in green rather than pink. So it's this stroma is actually no different between the testis and the ovary at this point. When we do single cell RNA-seq, they have a similar identity. What's different is actually what lives inside of these epithelial follicles. So this is the Sertoli cells of the testis and the male germline. So this would be the granulosa cells of the ovary and the female germline out here. To understand where human ovaries, how they form in development, we performed single-cell RNA-seq and published these studies over the last few years of the developing human ovary. And so what we discovered are transcription factor signatures which demonstrate the progression from common progenitors. So these are progenitors that are found in both the XX embryo and the XY embryo, which would give rise to the ovary and the testis. So beginning with GATA2, NR2F1, and PDGFR alpha. And then as we move through the common progenitor state, we move into the specification of the ovarian cells, the pregranulosa cells, versus the specification of the Sertoli cells. So specific markers of the pregranulosa cells include LGR5, which is a friend to many of us in the stem cell community. It's found in tissue stem cells in the adult body, and it turns out it's found in early progenitors of the ovary, as well as TAC1. And then in the testis, SOX9 is a transcription factor that we know drives Sertoli cell differentiation. So it shuts off in the ovary, it turns on in the testis due to SRY, and that leads to the specification of the Sertoli cells versus a granulosa cell. So human fetal tissue research is really informative for instructing development, instructing what are the key markers and transcription factors we need to think about when we're building organs in the lab. But human fetal tissue research also has some challenges, and that is because when we can access human fetal tissue to understand the development of these organs, it's right at the point of ovarian induction. So that single-cell RNA-seq I showed you, it took years to accumulate ovaries at that age because it's very, very difficult to obtain them because we're right on the edge of where we're able to acquire ovarian or fetal tissue for research. And then the establishment of ovarian follicles, I have very few examples of this using human fetal tissue, tissue because it forms after 20 weeks of development. So that means we need to think about other organisms, other models to be able to study the formation of the ovarian somatic cells and the formation of ovarian follicles. So to do that, about a decade ago, we started the Primate Ovary Project. And so this is a collaboration. The current iteration of it involves us at UCLA with the Oregon National Primate Research Center, as well as collaborations with our colleagues at UCSF, Diana Laird's lab, as well as computational support by Jian Xu's lab at the Mass General Hospital. And we're using the non-human primate tissue to fill in the areas that we can't fill in with human embryos and human fetal tissue research. So what this includes is the specification of the ovary. This is actually an indifferent gonad. It looks very similar to a mouse, and then things look very different from a mouse after that. And so this is at around 34 days post-fertilization. And now this is a third trimester ovary in the primate. This is the breaking down of those follicles and the very first establishment of the ovarian reserves, those primordial follicles. So using these alternate species is the only way that we're going to be able to understand the development of these key functional cell types and entities in the ovary using non-human primates. Because this is a very precious resource that is will be made available without restrictions to the scientific community, we're doing a lot of single-cell RNA-seq that people can use in their projects, a lot of spatial transcriptomics, and we're also making pluripotent stem cells from the rhesus as well to be able to differentiate these organs in the primate. So now I want to talk about the ovarian assembloids. So this is very routine in the mouse. There are two types. You can both of them begin with germ cells made in vitro called primordial germ cell-like cells, and they're combined together with fetal ovarian somatic cells that come from a fetal ovary. At the mouse, it's E12 and a half of development. That's equivalent to around 6 to 7 weeks post-fertilization in humans. So very, very early, very difficult to access those tissues in humans. That makes something called an R ovary, a reconstituted ovary. The other approach involves no fetal tissue, but rather differentiating the fetal ovarian somatic cells in vitro from pluripotent stem cells. And these cells are called fetal ovarian somatic-like cells. So in the reproductive science community, we like to refer to things as like cells. What that essentially means is the cell type is made in vitro. So therefore, it's like the in vivo cell type, but it's not originating in vivo. So that's how we use the LC terminology. We use our ovaroid technology, which is entirely in vitro, in order to develop the principles for generating human ovaries entirely in vitro without the use of fetal tissue as part of the assembloid. But the classic approach for making these assembloids of the ovary entirely in vitro are using the fetal ovarian somatic cells from the embryo, combined with germ cells made in vitro. So this is what the process looks like. You combine the germ cells with the fetal ovarian somatic cells to make a three-dimensional aggregate. It's a very precise number of cells, 5,000 germ cells, 50,000 ovarian somatic cells, combine them together, and it's very disorganized. Over the next 2 days, they sit in a 96-well plate in submersion culture. Then we move them to the air-liquid interface where they grow like an organ culture, for those of you who've done organ culture in the lab. During this time, there is organization of the somatic cells and maturation of the germ cells. Between day 8 and day 15, we begin to have the oocytes forming within these in vitro organs, and then by day 21, that's the end of the culture. After this, our ovaroid will begin to collapse unless we start changing the media. So I'm going to tell you about that in some unpublished data. So to go through the germ cells made in vitro, these are early stage germ cells, very similar to the germ cells that we make in vitro from human pluripotent stem cells. So what this is showing you is in the mouse, we start with, in this case, it's an induced pluripotent stem cell line, which has reporters, which are marking stellar expression through CFP and blimp expression through venous expression. We do a two-step protocol through epiblast cells and then three-dimensional disorganized aggregates, again, similar in principle to the human system, and that makes these organoids, and you can see that the germ cells form as clusters, and they tend to form as clusters and bud off from these assembloids, which we find is very characteristic of this model. If you do single-cell RNA-seq of this assembloid, you can find that it's full of BMP4 responsive cell types because we're adding a lot of BMP4 to drive germ cell formation. The germ cells are seen here, and this is some characterization of these germ cells here. They have a pluripotency signature of OCT4 nanoxOX2. We know that. That's very specific to germ cells. And then they turn on their germ cell specification signature, including BLIMP and DPPA3. We have some determination, which are called late-stage germ cell genes. These turn on as the germ cells would start to approach the genital ridge in different gonad. They begin to express dazzle and vasa, and we begin to even see some transcription of this in our in vitro model. You can also appreciate the germline is a very small fraction of the overall aggregate. So the aggregate's mostly somatic cells, embryonic somatic cells. The germ cells are around about 5%. The ovarian somatic cells at E12.5 include specified ovarian cell types. So we make them from E12.5 mouse embryos, which we isolate something called the mesonephros and the ovary from these embryos. We then slice off the ovary from the mesonephros. We trypsinize that to single cells. We use a MAX to remove endogenous germ cells because those endogenous germ cells, if they're there, they like this niche, so they will tend to go and make the niches first, and the iPS-derived germ cells won't. So we need to deplete the endogenous germ cells to make space for the in vitro germ cells to form follicles, and then we cryopreserve them. So what we have is freezers of cryopreserved fetal ovarian somatic cells that we thaw to make off-the-shelf ovarian assembloids. So when we thaw them, this is what's inside those lots of ovarian somatic cells that we manufacture. They're composed of granulosa cells, epithelial cells, which line the surface of the ovary. I have my glasses on and I still can't read. We have the stroma cells, so the NR2F2 positive cells. We have some max escape germ cells, which we're actually finding are very helpful because in the one organ you have endogenous germ cells and iPS-derived germ cells. So they're few in number, but they do develop. So we can now do comparisons within the same organoid of in vitro oocyte development versus in vivo, as well as some blood cells, which ultimately will disappear. On the day we make ovarian organoids, we sort the germ cells using stellar CFP and BV, the double positive cells, the specified germ cells. We thaw out a lot of the fetal ovarian somatic cells from the freezer. We combine them together into a three-dimensional aggregate composed of in vitro germline and fetal ovarian somatic cells for 2 days, and then we transfer them to the air-liquid interface where they are very disorganized at the point of transfer. So these big vasa positive cells in blue are the endogenous germ cells, in yellow are the iPS-derived germ cells, and in pink are the granulosa cells. So there's no follicles at this point at all. It's just a disorganized array of cells. Over 21 days, that's when our ovary will organize. Those germ cells will mature into oocytes, and the granulosa cells will surround them to form the follicles. So here is showing you a side profile. So the initial ovary I showed you was a top profile. It's like a dinner plate. When you look at it from the side, it's about a follicle thickness. And these are the oocytes in blue, marked by vasa, the FOXL2 positive granulosa cells that are surrounding them. This is surrounded by a laminin basement membrane, so they're producing basement membranes around those granulosa cells. And we have NR2F2 positive stromal cells in between. So we've got the major cell types of the ovary that are associated with the formation of the follicle and what we hypothesize could be endocrine activity. The oocytes at day 21 are very predictable in size. They're around 50 microns in diameter, and so it doesn't matter which lot of FOSC we thaw out. It makes about 50 micron in diameter oocytes at day 21. One of the things we noticed in the stroma is we're making these secondary follicles. We should start to see recruitment of what's called theca cells, so stromal cells that become specialized to produce the substrate needed to make estrogen and progesterone. So we wanted to know could these organoids be used to model the production of estrogen and progesterone by the ovary? And so this is just showing you here that the production of these hormones requires communication between these thecal cells, which express 3-beta-HSD, which is part of the pathway for forming hormones in the ovary. The precursors to estrogen are sent from the theca to the granulosa cells, and that these granulosa cells will then be the ones, which are the FOXL2 positive cells, the ones that make the hormones. So in an ovary, this is a postnatal day 14 ovary, we can start to see 3 to beta HSD around follicles. We didn't see that marker in our in vitro system. So what we did in collaboration with Shou Xiao's lab at Rutgers University was to develop an add-on protocol to the ovarian assembloid protocol, I should say, where we supply cytokines that are secreted by the oocyte, because the oocyte is actually the weakest part of this model. Only 5% of oocytes are functional, but they do support the communication with the granulosa cells. So when we supply a little bit more oocyte factors like GDF9 and BMP15, and we continue to culture these organoids, the granulosa cells start to change in their identity. This is some real-time PCR we're testing out for changes in granulosa cells specifically, so the ability to respond to FSH, the ability to respond to LH, the ability to generate hormones by looking at enzymes involved in steroidogenesis, and then we measured hormone production in the media. And so what we were able to show is that through this extended protocol of these in vitro ovaries, we're able to get really good production of testosterone, estrogen, and progesterone by these models of the ovary. So not only are we now able to model the assembly of these follicles, but also hormone production as well. And then you can start to imagine that this can help us to understand what can modulate hormone production, what in the environment can modulate hormone production by granulosa cells by screening through these ovarian assembloids. So I want to end now just in the last minute or so talking a little bit about human ovarian assembloids. I have no data to show you because all we've done right now is with making the individual cell types. I've shown you that we can and the field can make human germline cells, and we use them to model development, but we will also use them to put into these human ovarian assembloids to make follicles. We're just starting to make human ovarian somatic cells now. We've been building the reporters. This is, and also developing the reference maps, because it's hard to make a cell type in culture if you don't know what its identity is. And so that's the purpose of going through making reference maps with human tissue, filling it in with non-human primate tissue where we can. But we're in the process of using that knowledge now to make cells. What I wanted to spend just the last 2 slides talking about is choices of iPS cells. When I was in Renee's lab, and I showed you that beautiful picture from 2004, we worked with whatever we could. We worked with whatever would grow. We as a field were not great at growing human embryonic stem cells, the field, because there was very few reagents to do it, to make up everything ourselves, and there's a lot of variability in the system. That has been solved now, of course, with commercial medias, and now the standard practice for growing human pluripotent stem cells is very, very manufactured and routine. When I came to UCLA, I made a whole bunch of new human embryonic stem cell lines. This again was before there were iPS cells, in order to understand differences between lines. And what we noticed is that there is differences between lines. Some lines make lineages more robustly than other lines. We as a field are still trying to understand this diversity, but definitely exists. And I wanted to put this paper here. This was a really beautiful paper that was published a few years ago around the pandemic, that looks at common genetic variation that drives molecular heterogeneity in human iPS cells. And also, as part of this work, they showed heterogeneity and differentiation. So as scientists, many of us work with 1 or 2 iPS cell lines and do all our modeling in 1 or 2. But I think it's important that we take a step back and start thinking about how do we apply the technology we're building in the lab across multiple lines because of this heterogeneity that exists. We don't want to be studying disease and development in one person, which would be one iPS cell line. So because we're interested in ovarian health and wellness, and fertility and infertility and reproductive health and wellness, I started to think about what is healthy. In the banks of iPS cells that are available, what is healthy? So when I think about my embryonic stem cell lines, I don't think actually they're healthy, because they were acquired from the in vitro fertilization clinic. That means a person came into the IVF clinic because they were struggling having a family and they were struggling with fertility. So if I'm interested in modeling fertility, maybe I shouldn't be using embryonic stem cells. Maybe I should be using iPS cells. So well, how can I tell if iPS cells are from reproductively healthy people? So this is just a table that shows different banks and repositories from around the world and their geographic location and what they have in terms of control lines. And so this is the way control lines are described. They're either considered disease status normal, healthy controls, isogenic controls, healthy donors, apparently healthy, or normal. But what is normal when we're talking about reproductive health and wellness? And so in all of these cases, the reproductive health of the donor is completely unknown. So what we've been doing at UCLA is going back and deriving new iPS cell lines from individuals of known reproductive status so that we can use these in studies moving forward to build models of the ovary and also the germline. And so one of the papers that we published during the pandemic was a set of lines that we derived from monozygotic twins, where one twin was normal and healthy and her twin sister had primary ovarian insufficiency, in order to understand early development of the germline in these individuals. And with that, what I've told you about today is that the major cell types in the ovary originate from different germ layers, the germline and the ovarian somatic cells. That means if we're going to be modeling this organ in vitro, we need to consider the differentiation processes of two different lineages before we combine them together. And that this has been showed very effectively using the mouse as a model. I've shown you that human germline specification can be modeled very nicely using human pluripotent stem cells, and so we're doing a lot of discovery about genes involved in the early human germline that are different than the mouse, and that I think it's important to generate human pluripotent stem cell lines of known reproductive status of diverse genetic ancestry in order to build ovarian assembloids and model ovarian development, disease, and dysfunction. And so with that, I'd like to thank you very much for your attention. For those of you who came in the room today, I'm really thrilled that you can be here, and also for those people who are online. So I talked about a lot of work today from my lab. This is the current iteration of my lab, made up of fantastic graduate students, postdocs, undergraduates. We have bioethicists, philosophers that work with us in the lab as we think through the questions in utilizing stem cells, working with embryos and modeling the germline. And so these are some of my key collaborators and some of the funding that my lab right now is primarily funded by grants from the NIH and the Bill and Melinda Gates Foundation. And the cell lines we work with were funded by CIRM. So with that, I'd like to thank you.
Fantastic talk. I was very interested in the sexual dimorphism slide that you had between male and female reproductive organs, and wondered about sexual dimorphism in aging. So you showed that the ovarian decline occurs very sharply at 50. What about male aging and telomerase expression or other classic genes that tell us about longevity of a stem cell?
Yeah, thanks, Kat. That's a great question. I think what's so interesting when we're comparing aging of the testis versus aging of the ovary is that the germline in the testis derives from a stem cell population. So you can imagine that the aging of stem cell populations in any organ of the body, that the testis is experiencing the same thing. So what's known in the testis is there's clonal expansion of spermatogonial stem cells, and that with genetic studies of aging fathers, you can start to see clonal variants that are emerging in the germline. So it's a germline stem cell population. So there are men who are fathers way into their 70s and 80s. But what is now emerging is information that the health of children born by older fathers is different than the health of children born from younger fathers. And so there's a lot of investigation into why that potentially could be. So it's known that that the incidence of autism is increased from older fathers. And so could that be due to something in the germline that's changing with age? The ovary, of course, doesn't have a germline stem cell population, as I said. So once those follicles are gone, they're gone. And so it's a very different physiological aging process.
Great talk. And a little bit along the same lines of aging in the system, as you mentioned, the reproductive age is moving out, and one of the key consequences, as I understand, is more cytogenetic karyotypic abnormalities, trisomy, and so on.
Right.
So is there any way to model that, to understand that, using this type of system, or what is
Yeah,
known
yeah.
about that?
Yeah, so first of all, you're absolutely right. I didn't talk about the aging of the germline specifically, and it is known that the oocyte ages, that older women, there's an increase incidence of children born with Down syndrome or embryos that have trisomies, monosomies. You're absolutely right, and that's due to non-disjunction of the chromosomes. So when those oocytes are first formed and those primordial follicles first form, the chromosomes are arrested at the end of prophase 1. So essentially they're arrested without having their double-strand breaks fully resolved. The sister chromatids and the homologous chromosomes are still joined together by cohesion. So there's been a lot of effort on trying to understand how that molecular complex remains with high integrity over the next 40 years. And so what's known is that the integrity of those proteins that are holding together those homologous chromosomes and sister chromatids, it starts to break down over time, and that is what's. And then as the oocyte resumes meiosis, that's when you have this non-disjunction of those chromosomes. So there's a lot of effort that is using animal models themselves to understand that process of cohesion between homologous chromosomes in the oocyte. So how could the stem cell model this? It would be basically starting to model, I guess, the loss of those cohesins, the loss of those proteins, and how does that affect the oocyte in these systems. Also, we don't know a lot about the integrity of the oocyte in this in vitro model anyway. Maybe it's an aged-looking oocyte. I don't know. So I think there's so much to learn yet. It's unclear.
Thank you for a very illuminating and reflective talk. I'm really interested in your discussion of the in the ovarian models, the need to look at variation because a model will be from only one iPS cell, there's only one individual, and understanding that the role of variation. So if you're assembling, or assembling is probably the wrong term, but if you're gathering materials to start to look at the comparative studies, you're basically generating phenotypes for these developmental processes. And will there, can you use, or will it benefit from, how will it benefit from looking also at genotypes of these individuals to understand the framework that produces that phenotypic variation?
Yeah. So the reason why we started to, in addition to what is healthy, what is normal, as we were thinking about making iPS cell lines, what I found very compelling from the philosophers and the social scientists that we work with at UCLA is the fact that the majority of lines that we work with, that I've derived at UCLA, come from individuals of Caucasian ancestry, which basically means that I'm building research tools and research models that model development, potentially disease, within one ethnic group without thinking about others. So the reason why we're getting into using generating iPS cell lines is not necessarily to look at genetic variation and ovarian reserve. Other people are doing that, but rather, so we as scientists who are building these biomedical models, that we're not that we are not blind, we basically are not working with blinders on, so that as we're working with iPS cell lines, we're not working from one particular ancestry and one particular group. So that was a big part of the reason that we started working with iPS cells from different ancestries, so that we could be more inclusive in our research program of different individuals in different ethnic groups, but not necessarily individual genetic diversity. But there are others who are interested in that.
Great talk. I was going to ask about your single-cell transcriptome in the origin of the somatic ovarian cell. And you focused on, or you showed us the transcription factors, but I wanted to ask a little bit deeper about the non-coding RNA transcriptome, and also regions that would be the chromatin would be remodeled.
Yeah, yeah. So, so we haven't performed any of those studies. The reason why we chose 10x Genomics single-cell RNA-seq to do these studies was because that technology is so amenable to discovery, and also small numbers of cells you actually can put quite small numbers of cells into the 10x platform. So accessing this tissue is very difficult. We only get a few of them a year, and so we wanted to make as much discovery out of a small number of samples as possible. So we focused on transcription factors because we're interested in the instructions for how you make these ovarian somatic cells, and in 10x Genomics, we've just been focusing on the coding regions, but not the non-coding regions. So that would be interesting to look at. We haven't. We also haven't done any of the single-cell ATAC-seq accessibility, looking at changes in accessibility. We haven't done that. Others have, but that's not something that we've been focusing on. Yeah.
Beautiful, really exciting work. Was particularly struck by the self-assembly of the follicles in your ovarian assembloid. And I was wondering if you'd had a chance, well, we know that communication, right, between the oocyte and the granulosa cells is particularly important. And I was wondering if you'd had the chance to kind of look at formation of gap junctions or any of those other communications.
So no, we haven't, but we would like to, because these assembloids, there's only a few people in the world who actually make ovarian assembloids, and they're beautiful and reproducible, but they're technically difficult to get off the ground. So you need highly trained technicians and staff to be able to do it. And I'm in a really fortunate place to have a wonderful group who do it right now. But all of those studies need to be done, like looking at, when is that follicle assembled during this process? It actually has not been shown. And so we're starting to do those experiments now. This is where the spatial technologies are becoming really, really valuable in the discovery of these assembloids and how they're being built and how they're communicating with each other. So this is the future studies we would like to do.
Hi, thank you. Beautiful talk. As a placenta biologist and interested in trophoblast stem cells, I'm stricken by how all the transcription factors that you mentioned are also in common with the trophoblast.
Right.
So I was wondering if you've been looking at what are the epigenetics drivers that are driving the same transcription factor to make germ cells rather than trophoblast?
Yeah, because there seems to be a close relationship between the specification of the germline and specification of extra embryonic lineages, that we've decided to do this through live imaging, which is why we wanted to convert a lot of our platform into 2D so that we could look at the emergence of the germline and emergence of the amnion in particular. And so that's the work that we're doing right now in the lab. So once we can identify that common progenitor and then ultimately the two cell types, the amnion and the germline, then we're going to be in a better position to answer that question in a fulfilling way. I don't have any specific answers right now.
The questioner wants to know, have any of your insights into ovarian development given you any further insights into oncogenesis, ovarian cancer?
Oh, that's really interesting. It's a really important point, given how many women suffer from ovarian cancer. So no, because the ovarian cancer, it's found on the ovary, but it's thought to originate from a different organ. And so a lot of ovarian cancers are originating from cells within the reproductive tract that land on the ovary and then form a tumor. So we are working specifically with the cell types that making up the ovary itself. And at the current time, we're not making other reproductive tissues, but I could easily imagine in future that we would start to look at the interaction between other reproductive tissues, the uterus, the fallopian tubes, as well as the ovary.
Well, thank you so much. This was wonderful, Amanda. And let's give a great round of applause. And welcome back. Don't wait another 20 years before coming down.